Assalamu alaikum, dear students. You are welcome to my biology class. Today we will start a new chapter, biotechnology. But do you know what is biotechnology? Biotechnology is the combination of two words, biology and technology. Hungarian engineer Carl Iricky in 1919 first coined the word biotechnology. Dear students, you know what is biology? Biology is the special knowledge about organisms. That means biology is the science about living world, and technology is the application of machineries or tools in many sectors of our life. That means technology means a manner of accomplishing a task, especially using technical process, methods or knowledge. Now, can you tell me what is biotechnology? Biotechnology has many aspects. That means the application of biotechnology are huge. Among them, two methods are very common. One is tissue culture and other is genetic engineering. Today our topic is tissue culture. Tissue culture. Before tissue culture, we have to have idea about tissue. But do you know what is tissue? A group of cell having same origin, performing same activity. They are called tissue. So we can say that tissue means group of cell of same origin and performing same activity. But culture is a method of multiplying living part. In this case, multiplying tissue of organism by letting them reproduce in predetermined culture medium under controlled laboratory conditions. That means tissue will be cultured in culture medium and the process of that is called culture. Now tell, what is tissue culture? I think now you will be able to tell me what is tissue culture. It's a cell, basically plant cell, and this one is tissue. Why this one is tissue? Because here are group of cells and at the same origin and they performing same activities. So this structure indicates plant tissue. Now can you tell me what is the figure? What is the third figure indicates? Now, if you want to perform tissue culture, then you need some materials. Number 1, explants. Number 2, mineral nutrients, vitamins, phytohormones, sucrose, condensing substance agar, glass container, a cotton plug, and an autoclave machine. Explants. What do you mean by explants? Explants is the parts of plant which are to be cultured, and explants is collected from mother plants, mineral nutrients. And you know, mineral nutrients are of two types, macro and micro. Plant takes these nutrients from soil basically, that's why these are known as mineral nutrients, vitamins. In the life cycle of plants, plant also use vitamins for their growth. Phytohormones, it's very important for plant, and the plant hormones are known as phytohormones. That means phytohormones play a very important role in growth and development of plants. Sucrose, it is to be used to prepare culture medium. Condensing substance, agar, is also used to prepare culture medium. Glass container, in this container culture medium for tissue culture is to be prepared. A cotton plug and an autoclave machine. The culture medium then is to be kept in autoclave machine for sterilization. Now the steps of tissue culture. Which steps are to be applied if you want to do tissue culture? Now, step 1, selection of mother plant. Step 2, preparation of culture medium. Step 3, establishment of sterilized medium. Step 4, transfer in root developing medium. Step 5, transfer to natural environment. Now step 1, selection of mother plant. For tissue culture, selection of mother plant is very important. And now step 2, preparation of culture medium. After collecting tissue, that means explant, this tissue is to be kept in culture medium. Establishment of sterilized medium. Prepared culture medium is to be sterilized, keeping in autoclave machine. Transfer in root developing medium. This step is to be applied in tissue culture after a certain time. Transfer to the natural environment is the last step of tissue culture. Now we will try to get idea about tissue culture from this figure. Dear students, it is mother plant. That means we will culture the tissue of these plants. That's why it is called mother plant. Then from mother plants we will collect explants, and as I said before that, explants, what do you mean by explants? Explants, the part of plant which is to be cultured in the process of tissue culture. And as explants, pollen, root, bud, etc. are used. Then you have to prepare culture medium. Firstly, you have to take a glass container, and in this container you have to keep some mineral nutrients, vitamins, phytohormones, sucrose, and to bring this culture medium in semi-solid state, you have to add agar, condensing substance, agar. Then after preparation of culture medium, you have to close the opening of culture medium with a cotton plug. Then we will keep the culture medium in an autoclave machine, and when we will keep the culture medium in autoclave machine, we have to set 121 degrees Celsius temperature, 15 pound square inch pressure, and we will keep the culture medium in autoclave machine for 20 minutes for sterilization. After that, culture medium is to be kept under room temperature. Now we will keep the explants, that means the tissue, into the culture medium. after some days, from these expellants, callas or plantlets will be formed, and these plantlets will have these plantlets will have shoot. That means that stem, leaves, but there will be no root. For the development of root, these plantlets is to be transferred into root developing medium. This one is root developing medium, and in this medium, from these plantlets, roots will be developed, and after that, in final stage, that means in final stage, these plantlets is to be transferred in natural environment or field level for adaptation. And this is all about tissue culture. Now I will summarize the process. Firstly, we have to take mother plants, and from these mother plants will be disease resistant, that means healthy, and explants will be collected from these mother plants for tissue culture. Then we will prepare culture medium with the mineral nutrients, vitamins, phytohormones, sucrose and agar. Then we will keep the culture medium in an autoclave machine for sterilization, and bring out the culture medium from autoclave machine. We will keep it room temperature for some time, and then we insert explants into the culture medium. After some days, the plantlets or callas will be grown from the tissue or explants, but these plantlets will have no root. For developing root, these plantlets will be transferred into root developing medium, and after a certain period, root will be developed and the plantlets will attain a certain height. Then we will transfer the plantlets into natural environment for adaptation. And dear students, I think you have get a clear idea about tissue culture, and it's a very important process in the field of agriculture. So that's all about tissue culture. Thank you, students. Assalamu alaikum.